So we'll get started. This is the plant structures and growth PowerPoint. I tried to put some of the highlights of this lecture that we're going to see. We're going to compare the differences between the shoot system and the root system. I didn't leave enough room to write this down, but we'll talk about the functions and the structures of the shoot system, also the structures and the functions of the root system. We're going to separate primary versus secondary growth. And the main thing that we're going to see on our lab activity is the different types of leaves as well as the leaf arrangement to the stem. I wanted to bring in a bunch of examples of this, so that's where we get our introduction to native plants. I think I was able to find about 15 or 16 plants, but only 11 of them made our native plant lab list. I was only able to find 11 lab quality specimens of those. So just a quick distinction, the stuff that we'll see in terms of native plants and whether the native plants have simple leaves or compound leaves, or whether they're arranged alternately, oppositely, or in a whorled fashion around those plants. Those are all the types of things we're going to see in the post-lab quiz. For the lecture quiz, that's going to be anything we talk about from the lecture. So shoot system versus root system, primary and secondary growth. Whatever we don't finish today, if we don't get all the way through that, there might be a few more details that we'll add about plants on Wednesday. And I mentioned it last week, but we didn't have time at all to do it last week. And we won't have time to do it today because we're getting the lab activity out of the way. But on Wednesday, I'm going to bring in a case study that we're going to work through in groups. And you'll get 20 bonus points, up to 20 bonus points by doing this class activity. It's going to be something that I pass out. You're going to break up into groups and you're going to answer some simple questions. They're the types of questions that I would get into my email inbox. And you're just going to have to use your resources like the internet and your partners, and you're going to think critically and come up with how you would respond to these questions. The whole thing might take an hour, but since we won't have a lab activity and not very much lecture, we should be able to squeeze that into Wednesday. So if you want those 20 points, try and be here for that. That's about as far as I can think into the future. I think past that, it's just review and whatever campus plants we can find a week from today. On the other side of the board, we've got our 4 major plant groups that we were talking about last week, and around the room, we've got examples, visual examples of all 4 types, the bryophytes, the pterophytes, gymnosperms, and a whole bunch of these different types of angiosperms. So if we're ready to jump into new lecture slides, I'll get going with this slide, and I'll just kind of duck myself out of the way here. Okay. So right at the top, we're going to compare shoot system versus the root system. And you can think of the root system as the part of the plant that's below ground. The shoot system is just the part of the plant that is above ground, and they have different structures and different functions. We'll start with the root system because there's maybe less going on down there. Maybe that's debatable. I shouldn't even say that. There's different things going on down there. The root system, you'll notice, doesn't have any of the green photosynthetic cells. Obviously, it doesn't have any sunlight down there, so it's not going to be making any of the sugars. Instead, we're going to see cells down there that are specialized for absorption of water and minerals. If we look closely at that root system, we can see 2 main functions. One, like I just mentioned, absorption of water and minerals and transporting those water and minerals up towards the leaves, the shoot of the plant that is going to be doing the photosynthesis, that does need the water, does need the minerals, doesn't have access to them, has to get conveyed those water and minerals by this root system. So conveying water and minerals, that's one. The second thing is anchoring this plant to the substrate. Some roots reach further down into the soil and are better at anchoring plants to substrates. You might have heard of certain trees that have shallow roots are much more likely to topple over in a windstorm, and you wouldn't want to build a house next to those types of trees. So roots function as anchoring the plant to the substrate and conveying water and minerals up to the shoot system. Here are some of those specialized structures, those root hairs that drastically increase the surface area on the tips of those roots. That helps pull in more water and minerals. But we also mentioned something else last time that those roots tap into that's not even a plant, that really helps convey water and minerals to the plants. What was that? Think back to Unit 2. I wish I had more coffee. This would be a good coffee break. What's something that root might tap into down there that's not a plant that would help it absorb water and minerals? Also something we talked about in Unit 2. Catherine?
Fungus?
The fungus. The fungus has the feeding body of the fungus. It was a collection of a bunch of those individual hyphae. It was like if a piece of spaghetti was a hyphal thread, then a bowl of spaghetti would be that mycelium, that collection of hyphae. And we said underground, it fed on dead roots and dead plant material, and it acted like a sponge, and it held on to water and minerals. And so plants that tapped into that mycelium had access to water and minerals when other plants that weren't tied into a fungus, they would dry out. So it makes it sound like, there is an advantage to being tied into a fungus. Like we said, you get water and minerals more than surrounding plants would. But what was the disadvantage to having your roots tied into that fungus? Yeah, throw it out. What do you got? Yeah, like 25% of the sugars that those plants produce have to go back down and pay off the fungus. So it's a symbiotic relationship. You could argue that the plant probably makes 25 to 30% more sugars than it would if it didn't have access to the fungus at all. So the fungus really kind of pays for itself in that sense. Okay, let's jump to the shoot system. This is the above ground part of the plant. And we're going to see the structures are different as well as the functions. We obviously see green photosynthetic parts to the plant. But then there are also those showy parts to the plant that are not about photosynthesis at all. Instead, those are about tricking pollinators. And maybe I shouldn't say trick. They do pay off some of those pollinators with a nectar reward. But oftentimes it comes with, some type of trick of dusting pollen on them somehow, getting them to carry that pollen to a specific target on the other side of the forest so it doesn't have to waste pollen. So functions of the shoot system we can say are basically twofold as well. One is going to be for photosynthesis, production of sugars. Kind of tied into production of sugars is transport of those sugars around the plant. So like we just talked about, sometimes the plant has to pay off a fungus down in the roots. And we will see that the types of vascular tissue in plants that carry the sugars, they're two-way streets. So the sugars can flow down back into the roots. They can flow over into a branch that's producing apples and can store that excess sugar in apples. Sugar can be transported all around the plant. And then in addition to photosynthesis, producing these sugars, the other function we can task with the shoot system is reproduction. That's where the flowers come in. In addition to, this is just a list of some organs of the shoot system. We said there are leaves. Those are the sugar factories. The flowers are there only seasonally and are part of the reproductive life cycle of the flower. So their function is reproduction, the leaves' function is photosynthesis, and the stem's function is to basically elevate the leaves and the flowers high enough in the, above the ground so that the flowers can be seen by a pollinator and that the leaves can have access to sunlight and can be efficient in photosynthesis. So there we can see the stems supporting both the vegetative part of the plant, this leaf, and the reproductive part of the plant, the flower. Back to this picture. We talked about the root system and we talked about the shoot system. We can see the shoot system is broken up into these vegetative parts, which are only leaves, those sugar factories. And then there are the reproductive parts that are holding up the flowers and eventually the fruits. We recognize this thing as an angiosperm because it's got a flower right off the bat. But another review question, do we know what type of angiosperm this is? We talked about monocots and dicots last time, and there's at least 3 clues on here that tell you what type it is, either a monocot or a dicot. What is it, and what are the clues that you see? What are those? Yeah.
How many petals are on the flower?
Boom, that's one. We got floral parts in multiples of 5 in this case, so floral parts in multiples of 4 or 5 told us that it was what type?
The veins and the leaves.
And the veins and the leaves are branched. They are not running parallel to one another. There's a third clue.
How many of the yellow things are inside the flower?
That would also be multiples of 4 or 5. That's right. But there's another one down here in this below ground part of the plant.
Like whether or not it has a tap root?
That's right. This thing has that main tap root and then has a bunch of lateral roots off to the side. It's kind of like the same way the vascular tissue branches in the leaves. See how there's a main vein down the middle of the blade, and then there's a bunch of lateral veins that run off of that. So the vascular tissue kind of looks the same below the ground as it does in a leaf. And there was another one that we listed that was the embryonic seed leaves, which are not present on this adult plant. So you picked out the 3 clues that we could see, and those 3 clues told us that we're looking at which one of those 2?
Dicots?
It was a dicot, that's right. So monocots were things like grasses, and the veins would all be running parallel to one another. I'm trying to think. Looking around, I actually did not grab an example of a monocot in here when you see your angiosperm. So there's lots of dicots. I'll do better next class period. I'll try and bring in some monocots too. Okay, so that's right. We're looking at a dicot. We saw several examples of that. Let's look more closely at these leaves here and how this plant grows. I'll point out our key to knowing whether or not we're looking at a simple leaf or a compound leaf. Like, where do we know where the leaf ends? And it has to do with just these main 2 parts to the leaf. There is this blade to the leaf. Sometimes there's just a single blade, and other times that blade is going to be broken up into leaflets. Here's an example of what we're going to call a simple leaf. It's got just one blade, and then there's that little stalk that attaches the blade to the stem. This attachment piece is called the petiole. Here's the clue. If you can find the petiole and where it attaches to the stem, what you'll find right in that little crease is an axillary bud. That little crease is called the axillary region, the armpit region, and there you're going to find a growing point on the plant called an axillary bud. This leaf here is broken up. This is a blade that's been broken up into multiple leaflets. But if you look at the base of those leaflets, there will not be an axillary bud. You'd have to go all the way down to, this would be the petiole, and I've ripped it off of the stem, but right there at the base of it, you used to be able to see a little axillary bud. That means this big thing that we're looking at here, there isn't an axillary bud at the base of any of these leaflets. This is what we're going to call a twice pinnately compound leaf. This whole thing is a leaflet that is itself, the leaflets are broken into even smaller leaflets. So the axillary bud would have been way down here at the bottom. I know me holding it up isn't sufficient, so we're going to break into a little class activity after this so that you can see the different types of leaves. But how we know whether it's, where the leaf ends is we're looking for this axillary bud. What that axillary bud is, it's a growing point on the plant. So let's talk about how the plant grows for a second. On our list, we've compared the shoot system and the root system. We know the functions of the shoot system and the functions of the root system. So let's jump into primary growth versus secondary growth. And we're looking at those, what we're calling axillary buds. These are at those little armpit regions that allow us to identify individual leaves. And then also at the tip of the shoot, at the very tip of the shoot, there's what's called an apical bud. So axillary buds in the armpit, and apical bud at the very tip of the shoot. These axillary buds and apical buds, they're part of primary growth. Let's add some details about primary growth. Primary growth we can see is done by apical meristems and lateral meristems. Apical bud and lateral bud are other ways of describing this. So we'll say apical meristems, and I'm going to squeeze this in here, and axillary, what we'll call lateral meristems. And meristems is just another term for bud. These apical meristems and lateral meristems, you can think of primary growth as adding length to the plant. So let's say these apical meristems and lateral meristems allow the plant to grow in length. So these allow plants to grow in length. That is that their shoots grow taller and their roots grow further into the ground. Furthermore, we could say that all plants are going to have primary growth. So this is something all plants do. Annual plants are plants that aren't going to make it through the winter because they don't have any woody parts. They too have, all plants are going to have what we call these apical meristems and lateral meristems. Let's look at them a little bit more closely. The apical meristems, we said, are found at the tips of the shoots, and the axillary, what we call lateral meristems, are found in those little armpit regions where the petiole attaches to the stem. Here's a picture of one of those things zoomed in. That's the apical meristem, the tip of the shoot. These are those lateral meristems, what we call the axillary buds or lateral meristems. Those are little growing points. So right there is where we're going to have cells that are undergoing rapid cell division. They allow the plant to grow in length. Like we said, all plants have these. There they are down there in the roots. We tried to look at one of these things last semester when we were trying to find cells in different stages of mitosis. It's really the explosion of new cell growth that is the engine that thrusts this root cap further down into the ground. Those cells divide and then they elongate, and that's really the push that pushes those roots down. So any of this growth that's happening in length at the tips of the roots or at the tips of the shoots is lengthening the plant. This is primary growth, so all plants have this. However, there's another type of growth called secondary growth. Not all plants have this. Let's get another color. We can say secondary growth is going to be something found only in woody plants. So woody plants only. When we're thinking about secondary growth, this is done by what are called vascular cambiums. So vascular cambium and cork cambium are going to be growth structures that we see on secondary growth. They're going to allow growth in width. I spelled that right, but growth in width is something that not all plants have, but it's going to allow these plants to put on multiple layers that's going to allow them to survive the winter through the freeze. So secondary growth is something that you're going to find on perennials. Annuals will only have apical meristems, where perennials are going to have some of this secondary growth. If we look at this vascular cambium, here's what it looks like. And I'll draw your attention just a cross section through the stem. If we could just peel away that bark layer, this outer surface is what we would end up referring to as the bark layer. Just underneath the surface of the bark, there are going to be layers of vascular tissue that carry the sugars. Maybe we should just go to this real quick to remind us that of the vascular tissue in plants, the xylem is what carries the water. The phloem is what carries the sugar. In this slide, the vascular tissue of the xylem and the phloem have been colored in a purple color so that you can see how this vascular tissue changes from the roots. It's in the very center of the roots. And then that vascular tissue kind of splits when we get into the stem, and it forms these rings that I should say it in a different way. I should say when we get into the stem, that vascular tissue is broken up into bundles. And those bundles form a ring that kind of go around the perimeter of the stem. There's a hollow space in the center where there isn't any vascular tissue. It used to be in the center in the root, but now it's no longer in the center in the stem. It's kind of broken up into these little bundles that make a loop around the inside of the stem. Then when we get out into the leaves, we can see that the vascular tissue is going to be branched. So that once again tells us that we're looking at a dicot. So xylem again carries water, phloem carries sugar, and when we go back a couple of slides to look at how those vascular tissues are laid down, they are laid down on either side of a vascular cambium. I mentioned this before. I said if we could peel away the bark, there is a single layer of living cells underneath the bark that's called the vascular cambium. And it's that single layer of cells that's responsible for laying down new phloem and new xylem. In fact, if we look at the picture down here at the bottom, what we can see, the C represents that single layer of cambium cells. These cambium cells are going to replace themselves, so there's always a single layer of cambium cells just underneath the surface of the bark. To the inside of this cambium layer, they're going to lay down new xylem cells. So the first year, there's the first layer of xylem cells that are laid down. And then the second year, there's going to be a second layer of xylem cells laid down on top of that. New xylem is always laid down towards the middle layer of towards the inside of the cambium layer. And you'll notice just to the outside of the cambium layer is where phloem is laid down. There's a layer of primary phloem, that's the first layer of phloem that's laid down. And then just on top of that, there's a layer of secondary phloem. And the third layer, there'd be a third layer of phloem, a third layer of xylem. Xylem is always laid down to the inside of the vascular cambium. Phloem is always laid down to the outside. Here's what's interesting though. As a plant is going to grow in width, what starts to happen is this layer of phloem, it starts to crack and split as the tree's diameter increases. And then we're going to see that there are little nicks that are filled in the way that phloem breaks. It's one of the things that's characteristic about different types of plants is how the bark grows on plants. Maybe when we take our campus tour of plants, since we don't have a lot of leaves to look at, I can share some of the characteristics of bark on plants. You can sometimes tell what species of plant you're looking at just at how the bark forms. And I'm saying all that just to say that the layers of secondary phloem are what crack and become the layers of bark. Just to the inside of the bark, you're going to have this layer of vascular cambium. And if we could see inside that, so here's that living layer of cells, that vascular cambium, just to the inside of the bark. The bark is all the old... Is that a question? Are you just stretching? Okay, cool. The bark is all of that old phloem that has been cracked as the tree's diameter has increased, and we can see just to the inside of this vascular cambium, this is all of the old xylem. Xylem was just carrying water, so the oldest xylem is right in the center of the tree, and it hasn't carried water in a really long time, so it's all dried out. This core of the tree trunk here is called heartwood because it's typically very hard. It hasn't carried, it's lost all of its moisture. As compared to the layers of xylem that are closer to the vascular cambium that have had water running through them more recently, this could still be filled with some moisture. So it's called the sapwood rather than the heartwood. This is all dead cells. So we mentioned one of those things in Unit 2 where we saw a fungus can come in and basically a fungus eats dead plant cells. So a fungus could hollow out the heartwood of a tree and not hurt it at all. Instead, it's converting all of these dead cells of the tree into fertilizer that drop back down into the ground and support a boost of growth in the old tree's age. So we're just looking at secondary growth here, these woody layers of xylem to the inside of the vascular cambium, and then the bark layers of phloem to the outside of the vascular cambium. Back to the vascular tissue real quick. I'd mentioned we've got xylem and phloem, and it changes where it's at in the plant, whether we're looking at the root, the stem, or the leaves. So far, we've just focused on vascular tissue, but I'll point out that there are two other types of tissue that we can be aware of. There's the dermal tissue that's shown in blue. Dermal tissue is like your skin. It's what protects you from the outside environment. And the dermal tissue on plants protects the plant from the outside environment. We'll see that a lot of the dermal tissue on plants, when you make your way around the room and touch some of these leaf surfaces, you'll see some of the dermal layers have accumulated a waxy cuticle. And that also just helps protect them from drying out. So the dermal tissue is the protective exterior of the plant. We talked about the vascular tissue carrying water and sugar. So in between the dermal tissue and the vascular tissue, that leaves only the ground tissue to talk about. And I think of the ground tissue as almost like the packing peanuts of plant tissue. You can see it's basically in between the vascular tissue and the dermal tissue in the roots. It fills the space between the vascular tissue and the dermal tissue in the stem, and it fills the space between the vascular tissue and dermal tissue out in the leaves. It just, again, is the packing peanuts and fills the space between the other two types of tissue. It has some functions, which we'll talk about, I think, in a couple of slides. Here's when we take an actual cross-section through a root and see that when we look at I don't expect you to really remember this, and now I'm reminded I got stung by a bee right here yesterday, so I've got kind of a little horn, but I should try not to scratch it, otherwise it'll get worse. Anyway, if we were to take a cross-section through the root right here and look at it under the microscope, this is what it would look like, but here's the actual picture. There's all of the vascular tissue in the center of the root. The dermal tissue is this thin layer around the outside, and in between this dermal layer and the vascular layer, there's all of the packing peanut stuff. This is what we've just called the ground tissue. If we zoom in on that vascular tissue, it forms this X, which might help you remember xylem is the tissue that forms this X. The structure we're looking at, this X structure, that's the xylem tissue with phloem kind of nestled in the spaces, in the little spaces of the X. Look how the phloem cells are not as large in diameter as the xylem. The xylem has to carry large amounts of water up the plant. Not as much sugar has to go back down the plant, so we can see the phloem tubes are smaller than the xylem. Xylem tubes are always going to be bigger than phloem tubes. This structure that we see with this X shape of xylem cells in the root is called the stele. Maybe just know that the stele is something that you see in the roots, and we don't see this anywhere else. In fact, when we get up into the stem, there isn't the vascular tissue in the center. Instead, we now see this vascular tissue is arranged in these bundles that have formed a ring around the perimeter of this stem. If we look at just one of those vascular bundles, I'll first point out that darkest staining tissue, that dark red staining clump of cells, that isn't xylem or phloem. That's a group of cells called Sclerenchyma cells. Those you can think of as, if we were to pour like a column of cement, to give that column of cement some additional strength, we would put a bunch of rebar in there. And those bars of steel would give that column of concrete some strength. And that's what these columns of vascular tissue of xylem and phloem, they need some kind of strength, some type of support pillar. And that's what these Sclerenchyma cells are. They're full of a bunch of lignin, a bunch of fibers that give them some tensile strength. They're real stiff cells. And that basically is the support that allows, right next to those Sclerenchyma cells, the vascular tissue of xylem and phloem to run right alongside the pillars of support there. Notice how xylem, like we mentioned, is always towards the middle part of the stem, and phloem is closer towards the surface of the stem. Phloem, in this case, is closer to the Sclerenchyma cells. Xylem is always more towards the middle. So vascular tissue is arranged in bundles when we look at the stem, and then when we get all the way out into the leaves, we can see that vascular tissue is once again going to be split. Right now we can see branching in the vascular tissue, so we know we're looking at a dicot. That vascular tissue, once again, the larger diameter vessels are the xylem, the smaller diameter vessels are phloem. But the other cells, we talked about the surface of the plant, this is the dermal layers. We can see that these layers of dermis have accumulated that waxy cuticle that I mentioned that help waterproof them. So it's difficult for water to move in and out. It's even difficult for gases to get across that waxy layer, which is why there are these little openings that we called stomata. That opening is the stoma. that when it is open, gas can easily move across that surface. It's what happens when I'm up here breathing. Gas exchange is occurring. So breathing is occurring across that stoma. It's just the opposite of what happens in us. When we're exhaling, we're getting rid of carbon dioxide and pulling in oxygen. What's happening in the stoma is it's exhaling oxygen and breathing in carbon dioxide. So gas exchange, water, prevention of water loss, and encouragement of gas exchange is what's happening across the dermal layers. Notice at the bottom surface of the leaf there are often twice as many of those stomata as there are on the upper surface of the leaf. That's just to help prevent them from losing excess water. So we talked about the dermal layers, we talked about the vascular tissue. Let's talk about the packing peanuts real quick, which is all of this ground tissue in between. You'll notice that on the surface of the leaf, the upper layers of ground tissue are broken into these columnar shapes. This layer of columnar-shaped ground tissue is called the palisade mesophyll. What's unique about the palisade mesophyll as opposed to this spongy mesophyll down here, you can see the spongy mesophyll, those are round cells, they are not columnar-shaped. Since they're round, there are these little gaps, these little empty spaces that fill up the lower surface of the leaf. That's where it gets this name of spongy mesophyll, all those little spaces that are in there. What fills up those little spaces, you'll notice those guard cells that open and close that stomata are more prominent on the lower surface of the leaf. In those little gaps inside those spaces, that's where oxygen makes its way through these guard cells and just hangs out in those little spaces. Waits until it's, let me try this again. I just said oxygen came in that guard cell. What I meant to say, carbon dioxide comes in through these guard cells and it hangs out in those spaces. It's going to be used whenever those cells start to crank through photosynthesis. In fact, anywhere you see the little green chloroplast, all of these ground tissue cells are going to be pumping out carbohydrates. They're going to be doing photosynthesis. As long as they have sunlight, as long as they have water, you can see each of these cells has that central vacuole that's filled with water. So those chloroplasts have sunlight. They've got water. They just need carbon dioxide. And that's where the carbon dioxide is hanging out and waiting to be used inside those empty spaces of the spongy mesophyll. When oxygen is produced, oxygen also starts to accumulate in those little spaces and makes its way out those stomata. So again, gas exchange occurs through those stomata. In these little gaps of the spongy mesophyll is where that gas builds up, carbon dioxide and oxygen. The columnar-shaped cells, this palisade mesophyll, the reason they're columnar-shaped is to be more efficient at capturing those wavelights, more efficient at capturing wavelengths of sunlight. If they were more squamous-shaped, flat, pancake-shaped, sunlight would easily penetrate straight through them. But in this columnar shape, they catch more wavelengths and are more effective at producing sugars. The lower surface has to have those gaps, those spaces in it to fill up with the gases of oxygen and carbon dioxide when photosynthesis is occurring. So we've seen ground tissue, we've seen the vascular tissue, and we've seen the dermal tissue. There are just a couple of pictures of the stomata. That takes us then into talking about the last point, which is leaf type and leaf arrangement. We've got plenty of examples around the perimeter of the room. We'll start with this picture of just a simple leaf. How we can tell that we're looking at a simple leaf, we've got the basic parts of a leaf. There's the blade of the leaf, and then there's the petiole that attaches the blade to the stem. That little armpit region where the petiole attaches to the stem is what we call the axillary region. And we should find, at least on our lab examples, there's going to be an axillary bud right there that tells us right off the bat that we've identified a single leaf. This leaf has just one single blade, but we're going to see that there are multiple ways that blade can be broken up. What we're looking at here are still just a bunch of simple leaves. That's a single blade that's got a petiole that attaches it to the stem. There's another single blade with a petiole that attaches it to the stem. What you'll notice about this first plant example is that there is only, let's add a vocabulary term real quick. The term node refers to a place where a leaf attaches to a stem. We know the petiole is what attaches the blade of the leaf to the stem. So anywhere a petiole touches a stem, that's a node. In a type of leaf arrangement that we call alternate leaf arrangement, this alternate leaf arrangement can be described as just having one leaf per node. So each node there, we see just one leaf attaching to the stem. Maybe a side note, 75% of the native plants in Texas have an alternate leaf arrangement. So this is much more common than the next type of leaf arrangement that we'll see called opposite leaf arrangement. There are a few examples that I brought in that are opposite leaf arrangement plants. This is described as having 2 leaves per node. A node is where a leaf attaches to a stem. There are 2 leaves that attach to a stem at the same place. So this is an opposite leaf arrangement, as its name would suggest. But then there's a third option here, and one of the plants in lab is an example of this third option. It's called a whorled leaf arrangement. A whorled leaf arrangement can be described as having 3 or more leaves per node. 1 leaf per node is alternate, 2 leaves per node is opposite. If it's 3 or more leaves per node, it's whorled. How the leaf attaches to the stem is what we mean by leaf arrangement. How is that leaf arranged to the stem? The options are either alternate, opposite, or whorled. The next thing we need to see is the type of leaf. So I'll just real quickly just draw our attention to these bottom 2 pictures here, because that's what we just talked about. We talked about how these leaves are arranged to the stem. They're either arranged opposite one another in what we call opposite arrangement, or they are arranged alternate of one another, where there's just 1 leaf per node. So if we can just discount these 2 pictures, the rest of the pictures that we see up here are showing us leaf type. What type of leaf are we looking at? The first type of leaf that we see here is just called a simple leaf. And like I was holding up here earlier of this magnolia tree, this is an example of a simple leaf. It's got just 1 blade. You can see the petiole attaching it to the stem. And they've even zoomed in to show you the clue that we're looking at. Right where that petiole attaches to the stem, there's the axillary bud. That tells us we found a single leaf. This thing over here that I was just holding up is also a single leaf, but it's an example of a compound leaf. There are a couple of different types of compound leaves. So the first one that we see is a type of compound leaf. This just means that the blade has been broken up into a bunch of individual leaflets. If the leaflets are opposite one another, like we see here, this is what we're going to call a pinnately compound leaf. If there's just 1 set of leaflets, this is what we call once pinnately compound. This mountain laurel is once pinnately compound. There is another example. This plant, the common name is burning bush. And it's going to be an example of a bipinnately compound leaf, where even the leaflets are divided into leaflets themselves. The leaflets are pinnately compound. Tomato plants are another example of this. So twice pinnately compound or bipinnately compound, we'll see examples of these. And then in addition to being pinnately compound, there's another option called palmately compound. And these, you can see each leaflet is going to radiate out from the tip of the petiole, like your fingers would radiate out away from your wrist. There are some examples I'll bring in hopefully on Wednesday if plants bloom in time, but right now in lab we don't have an example of a palmately compound leaf. And then there are these little vascular bundles, these needle-like leaves. We found them on the pine tree. Just to make a distinction of, these are all types of angiosperms. And the last 2, either these scale-like leaves or needle-like leaves, these are indicative of gymnosperms. We do have examples. The juniper tree have scale-like leaves and the pine tree has needle-like leaves, but we wouldn't be asking things like opposite or alternate for scale-like leaves or needle-like leaves. Those are all going to be examples of questions that we might see on the angiosperms. So the lab list, like we said, when we go around the room, if you were to take a botany class, you would have to remember all of the different shapes that leaves can take. These are descriptive things. Here's a bipinnately compound leaf where you can see the leaflets are divided into, each leaflet is pinnately compound. There's a once pinnately compound, there's another once pinnately compound leaf, that's a palmately compound leaf. You can get close to see how the veins are going to branch differently in leaves. This is another distinguishing characteristic. Also, there's a couple of descriptive characteristics that we might see on Wednesday that allow us to distinguish the different types of simple leaves. So all of these are simple leaves, but in addition to having different shapes, they can have different edges on the leaf. The edge of a leaf is called the leaf margin, and sometimes those leaf margins are smooth, which is instead of just calling it a smooth leaf, the term that describes a smooth margin of a leaf is called entire. There are other terms like dentate that describe this jagged edge of a plant leaf. Serrated is another even more pronounced tooth on the margin of a leaf. There are lobes. There's all kind of varieties. Some of them have little spikes on them. And in fact, you'll see oak trees back there that have catkins, but they'll have different leaf shapes. Those are some of the distinguishing characteristics between the types of oaks. That's the last slide that we have, so I won't go any further than that. I'll maybe come back to these two slides because we're about to jump into our lab activity. I want to pass out a checklist that you're going to get. It has 10 spots on it. And I went ahead and accidentally added an 11th plant to the list because I found one on a good example in the parking lot. So you might have to create an 11th spot down there.